Good afternoon. Welcome to the third webinar from MBF Bioscience. My name is Susan Hendricks. As you can see from these pictures, we've received over 10 inches of snow and counting here in Vermont. We're hopeful that there'll be no power outages during this webinar. Should that happen, the webinar may abruptly cease and we'll have to reschedule. I'm really hoping that that doesn't happen, but I just wanted to warn you that that's a possibility. Today I'll be demonstrating how to perform neuron reconstruction using Neurolucida. At this time, I hope everybody can hear me, and if you need to, adjust the volume of your speakers. My voice isn't as loud as that test voice, so you may need to turn up the volume. The webinar will last 60 minutes and is divided into 4 topics. The goal for today's webinar is to provide practical demonstration using Neurolucida for neuron reconstruction. The data you require to test your hypothesis will ultimately drive how you use Neurolucida for cellular reconstruction. We'll briefly touch on the equipment considerations for tracing at the microscope. This will include a discussion and demonstration of some of the features of Neurolucida to make this process of reconstruction flexible. Finally, I'll display some of the analyses available in the companion program to Neurolucida Explorer. There'll be a short break after each topic where I'll address questions that have been submitted via the question window. If you have additional questions that are not addressed by the conclusion of the presentation, please post your questions to our newly developed user forum to stimulate discussion offline. This can be found at forums.mbfbioscience.com. Creating an accurate representation of cellular features can be used in a variety of ways to examine a multitude of experimental hypotheses. Just a few examples include terminal field analysis, where axons are traced across many physical sections, to the analysis of individual spines along dendrites, to the creation of a wiring diagram or connectome. In addition to morphometrics, the reconstruction can be used to qualitative demonstrate your point with 3D viewing. With computer-assisted neuron tracing, the user traces by placing points along a neuron, either manually or automatically. And this can be done in both 2D and 3D, on live or captured images, and from a single section or many. To create a realistic, meaningful, and quantifiable model of a selected neuron from a biological specimen, Neurolucida provides tools to allow you to place points that represent the location in 3 dimensions, each portion of the segment, spine, or soma, approximate the thickness of the axons and dendrites at each point, and specify and classify the spines on the basis of shape or head diameter. Editing tools allow the user to erase or add branch points, entire trees, and group selected objects for analysis. You can save images with the tracing or save them separately. The tracing itself can be saved as a DAT or an ASCII file and can be stopped and started across many sessions by simply realigning the tracing with the histological section. The method, accuracy, and thoroughness used in performing the reconstruction is dependent upon the analytical needs of the researcher. So a primary question is, why are you reconstructing the cells? Are you reconstructing one cell per section, such as in the example of using a cell that was previously recorded from and then filled with biocytin? Does it extend through multiple sections? Can you see the soma in the data that you're providing? And do you need that information? If you're working with a method that stains multiple cells, you'll need to select the cells to reconstruct and disambiguate processes from neighboring cells. Here are a few examples of commonly used visualization methods for reconstruction. You can use a stain that unselectively stains multiple neurons, like Golgi or genotypic GFP expression. You can use a specific stain that selects specific cells, such as intracellular injections, or use a method that selects populations with antibodies or some other method. You'll also select the appropriate imaging method for the modality that you need.
So I'm going to move on now to the equipment requirements. Many hardware configurations are suitable for performing your neuron reconstruction. Today I'll be performing the demonstration on a Zeiss Axio Imager Z1 microscope, which has a motorized objective turret and internal Z motor and encoder. A Ludal XY motorized stage will be controlled via a joystick and the live image feed will be sent to the monitor with an Optronics Microfire CX9000 camera. All the hardware will be controlled by version 9.10.1 of Neurolucida. We recommend using a computer equipped with the latest version of Neurolucida and dual monitors. The increased workspace afforded by the second monitor allows you to keep software features that increase your efficiency available during the reconstruction. Due to the webinar format, the demonstration today will only use one monitor. Finally, you need to use objective lenses that provide the needed resolution and depth of field. Kohler illumination should be achieved prior to beginning your reconstruction, since it will optimize optical resolution and decrease eye strain. This is, of course, not necessary for fluorescently labeled tissue. Neuron reconstruction can be performed on image stacks that were collected either from a microscope that has Neurolucida installed on it, or you can import image stacks captured from a different microscope. In the case of importing image stacks, it's necessary to ensure that the program is configured with the proper XY scaling of the lens and the Z step used to acquire the image or image stack. When tracing structures that extend across many sections, movements over multiple fields for XY coverage within the section is best achieved by working at the microscope, in my experience. Neurolucida can control the motorized stage to move the specimen under study when the area of interest is larger than the field of view. Here on the left, you can see a reconstruction of an axon's terminal field within the corpus callosum. It was reconstructed from 150 serial sections, but was traced to record the fine detail by creating the reconstruction at a high enough magnification. These multi-scalar reconstructions can be very instructive, and this is most easily achieved with a well-calibrated system. When I say well-calibrated, I am primarily talking about these 5 types of calibration. First and foremost, you should ensure that your camera is aligned with your stage. This ensures that your movements are accurate. The lens and Z motor calibration ensure that distances in X, Y, and Z are accurately recorded in your data file. Parcentric calibration keeps the tracing aligned in X and Y with the camera image as the objectives are changed. So as you move from 4X to 10X, Neurolucida will know where you are, what's visible relative to where you just were. The same thing is true with parfocal, except in Z. So this keeps the tracing aligned in Z with the camera image as objectives are changed. Finally, I can't stress enough the importance of Kohler illumination for optimal tracing when you're using bright field stained tissue. Let's move on to the data file so that we can get on to using Neurolucida. So I'm going to talk about setting up the data file in the workspace right now. Just take a couple of minutes before we go to the microscope. Okay, so you'll want to lay out your data file so that the data will be collected in a manner that's easy to analyze, align, and resume. You can create a contour of your region of interest. So if you're analyzing cells within the cortex, which is what we're going to be doing today, you can trace a contour that bounds the entire cortex for that section so that you have a reference for where that cell was inside your region of interest. Using fiducial points when you're tracing your contour will help with alignment later on. And I like to collect images of candidate cells. So as I'm looking at low power, I'll use markers to mark where cells of interest may be. All of these things I'm going to demonstrate, by the way. You'll collect images of these candidate cells as you're going along so that you have a record of cells that you think might be appropriate for analysis. As you're tracing, you can group structures as sets for analysis. This is especially important if you're going to reconstruct more than one cell per data file. And finally, you can plan your tracing to fit the time available. So if you only have an hour at the microscope, you can set up the goal for your tracing for that hour to make the most of that time. So that not only do you get the most out of that hour of tracing, but when it's time to resume, the data file is in a position to easily resume. There are a number of user-defined settings in Neurolucida. One of the things that we hear often about is what's the reference point and why do we care? The reference point is actually critically important for Neurolucida because it defines the coordinate space. It is 0, 0, 0 in XYZ for your data file. Everything else is drawn relative to the reference point. So if you're having trouble getting Neurolucida to do what you want when you first open the program, it may be because you haven't placed a reference point yet. Another feature that is commonly used, I'll toggle this on and off, is display thickness. This allows you, as you're setting thickness, as you're tracing, you can see it on or off. Auto move automatically moves the stage as you trace. And you can use the middle mouse button to define the segment diameter, or you can use it to focus. So you can control the Z motor. There are a number of toolbars that Neurolucida has available too, so you can dock these or put them on your second monitor, which I recommend so that you have a very large user space for tracing. But the Z meter allows you to see where you are in that section. As you focus up and down, it gives you a reading. The macro view allows you to see where you are relative to everywhere else in the data file. So this white dotted line here defines what is visible on your tracing window. The orthogonal view is something that the more you use it, the more you like it, I think, personally. The orthogonal view allows you to see the reconstruction through Z. So you get an XZ projection. That's what we have listed or defined here. And so you can see where you are in Z. And so you can see the changes in the structures as you move through Z. The serial section manager is extremely important if you are tracing a structure through many sections. This allows you to easily identify, and also helps with alignment as well. The image organizer is handy to keep track of those images that you're acquiring of candidate cells, or a virtual slice of your entire region, or an image stack, or all of the above. It allows you to toggle visibility on and off. The 3D visualization window has undergone dramatic changes for version 9, and I think it makes things a lot easier to use and view. So you now have the ability to have a YZ or an XZ projection, and you can slide the slider up and down to change the focal plane. Additionally, the 3D visualization module allows you to capture a video of this. It's very handy if you have a presentation that you're preparing for and you want to drop some of your reconstructions into your seminar talk. And finally, new for version 9 as well is the editing tool. So this allows you to dock this toolbar along the side of your tracing window, and you can then do multiple editing tool operations all one right after the other without having to go in and out of edit mode. And it makes things a little bit easier to follow. Okay, so let's get right to Neurolucida now. That's really what we're hoping to get into here. So Neurolucida should now be visible on your screen. There we go. And I have a live image. To adjust the camera or the video input, it will change depending on your camera. This is the setup for a CX9000 camera, and you can adjust the exposure settings. You can perform an automatic white balance and set any binning if you need to. I recommend that you set the exposure time less than 100 milliseconds. This is especially handy for placing points while focusing. It keeps everything real smooth. So try to adjust your exposure settings so that they're less than 100 milliseconds. We are looking at a section of Golgi-stained cortex right now, and it's also counterstained with a Nissl stain. And we're viewing it at 2.5x. You can see my Z meter, macro view, orthogonal view, and image organizer. These are all docked against my tracing window. And I'm going to turn on auto move. Aha! This is a key point right here. I don't have my auto move box, and that's because I haven't placed a reference point. It's a classic situation. This happens all the time. And so choosing your reference point it can help you when you need to resume your tracing. So it helps to pick the reference point in some place that is easily identifiable, whether it's the corner of the section, the corner of the slide, someplace that is very easy to see. And so now that I've placed my reference point, you can see that I have the auto move box available to me. And if I switch to contour mode instead of tree mode here, I'll be able to trace a contour to represent the cortex. So I'm going to trace a region of the cortex right now to give myself some workspace. And you can see that the macro view is automatically updating. The orthogonal view is updating as well, but I haven't made any adjustments in Z, so it doesn't look like much. So I'm making a rather arbitrary contour right now of the cortex. But what you can do is define the contours relative to your research question. So if you want to analyze structures within different layers of the cortex, you can define different contours to represent those cortical layers. Additionally, under display preferences, you can adjust the tracing transparency. This is also new for version 9, and this is extremely handy. This will come to the fore when we start tracing. But you can see here that now we can have display thickness on and adjust the transparency so that we can see the tissue below. This is super handy. Okay, so you can do a number of things once you have a contour that bounds your region of interest. You can do a meander scan through your entire contour so that if you want to identify candidate cells, you can make sure that you examine all parts of your contour. Additionally, you can just select markers and place them near cells that look interesting to you. We can use field moves if we'd like to move, or you can go into Joy Track to examine your tissue in greater detail, or with greater specificity, I suppose. If you want to change magnifications, you can do so. So we can go up to 10x. And now we can see the tissue a bit more clearly. And because I've done a parfocal calibration, and percentric, and the lenses are calibrated, you can see that the contour is still along the region of the section. And in macro view, if we right-click and select Go To, we can now drive the stage to the place where we place markers at the lower magnification. So you can imagine that this can be really handy when you're surveying your tissue, looking for those cells that are the ones that meet your criteria to be traced. So let's now go to 40x and look at these cells in a bit more detail. Okay. We may need to adjust the camera settings. And again, you want to keep the exposure time less than 100 milliseconds. Okay. And so here's the cell that we had marked before at lower magnification. It looked quite decent, but I would exclude this from my analysis. I wouldn't choose to reconstruct it because it doesn't look like there's some breaks in this Golgi impregnation. It doesn't look like it has survived the sectioning process all that well. So let's check the other marker so we can. think this guy will do. Yeah. Okay, so let's reconstruct this cell. So here we can see that it's got a couple of breaks, but overall it looks pretty good. You can use the criteria you need to in order to reconstruct your object. We're going to switch to contour trace, or from contour tracing to neuron tracing mode, and trace the cell body, or the soma, generally at the midpoint of the thickness of the soma. Oh, oops. I was tracing as a dendrite. To undo, you hit Control-Z, and we can select cell body and trace the cell body of our candidate cell here. We can switch now to dendrite and start reconstruction. And so I'm changing the circular cursor diameter by using the scroll wheel on the mouse. And as I do that, to choose my first point here for this branch here, I'm going to bring it into best focus and mark a starting point. And as I move along the tracing, or move along the branch, I'll adjust the circular cursor to match the diameter of the branch below it. We can go to Display Preferences and make the tracing transparency a little bit more transparent. And so now you can see the tissue a little bit better underneath. And as you're tracing, if you're reconstructing spines, if spines are of interest to you, you can use the spine marker tool if you'd like, and either set the head diameter of the spine using the circular cursor, or if you're really just interested in spine numbers and you're not interested in classifying them, you can leave the head diameter any size that you'd like. You can exit the spine tracing tool and continue tracing. When you get to a branch point, you'll right-click and select Bifurcating Node, again adjusting the focus as you come along, and then choose a branch to continue down. If you make a mistake setting the thickness, so you can see here that I set that thickness too big. If you hold down the Shift key when you place your next point, you can see that, can you see that point that I just placed? I'm changing the thickness of that as well. So if you hold down the Shift key, it allows you to adjust the thickness of the most recently placed point as well as the one that you're about to place. And so you can use that to continue on. And so reconstruction is actually decently simple once you get the hang of it. When you reach an ending, if you're tracing only within one section, you can right-click and select ending. You can also specify what type of ending. So if you are tracing between sections or among sections, you can specify a high or a low ending, which helps you figure out which way to connect the branches when you move to the next section. And so if we continue on, again, we'll just focus as we go. And pay attention to the ortho view. The ortho view is pretty handy in this way. We can also turn on the Z filter of the ortho view and change the range. So if we change the range to 2 microns, say, as we focus in and out of section thickness, you can see parts of the only parts of the branch that are within that range appear, which if you've got a real tortuous soma with lots of branches, this can be an easy way to sort of keep track of where you're going and what's been already traced. Keep your screen as clear as possible. So I'm going to pause right now just to see if there are any questions about this very basic part of tracing before we move on to other topics. Catherine has a question. She's interested in tracing spines, which based on the registration questions that I asked before the webinar, it sounds like a lot of people are. And her question is, can I trace the dendrites first and then mark the spines after? The answer to that is yes. So you can do, with the Neurolucida allows multiscalar reconstructions, and we try to make it as easy as possible. So you can reconstruct the branches at 40x and then switch to 100x to mark the spines. You just have to make sure that you're at the right Z level so that you have the branch in focus before you start marking those spines. But you can do them in sequential order if you'd like. That can be a great way to approach your experimental question. Kaylin asks the best way of finding the point of reference from my own experience. Personally, I like to place the reference point in a place where that makes sense to me for my reconstruction. I always tend to pick the longitudinal fissure if it's available, so the midpoint where the corpus callosum is. That seems to work for me. And then I typically, when I trace my contour, I like to have the contour as well because that allows me to align the tracing grossly pretty easily at a low power. It's pretty difficult to think about aligning the tracing of a neuron that you'd like to resume at low power. You typically have to be at a higher power. So placing the reference point and having that contour that I trace also as a reference. I make a fiducial portion of the contour extremely accurate so that I can quickly align. Oh, we have a question about can I set the dendrites with different colors? So can you trace the dendrites with different colors, and then when you're all done, can you train them to the same color? Absolutely. We automatically vary the color of the dendrites as you're tracing just to make the process of tracing a bit more straightforward. So you can see who belongs to whom as they begin to overlap and things like that. But I'm going to end this just so that I can demonstrate. If you'd like, you go to Display Preferences, select Neurons, and then you can also select here how you want to color the branches. You can have it color by branch order, or you can just set all of the structures to the same color after the fact, if you'd like. When you're tracing, when you place a node and continue tracing down a single branch, when you get to the end, it'll snap back to the node closest to where you were tracing and adjust the Z to bring you back to the same Z point of the point where you placed the node. This allows you to keep track of where you've been and what you still need to do. And I think it's quite a handy feature. Okay, I'm looking over some questions. You guys have a lot of great questions. This is fantastic. I hope you'll carry some of these questions over to the user forum so that other people can share their strategies as well as the information that I'm sharing now, which represents sort of how I like to do these reconstructions. There's been a couple of questions about having when you're tracing things moving or shifting and you have to realign the tracing with your structure. That is best solved by doing a very accurate calibration. So those 5 calibration steps that I talked about, revisit those. It's a possibility that you have slop in the turret for your lenses, which means that as you switch from position to position on the nose piece, the turret for the lenses, it doesn't seat in the same exact spot from switching to switching. There's not a whole lot you can do about that except get very good at aligning your structure. That is nothing to do with Neurolucida. It's an issue with your microscope. And okay, so let me trace a couple more branches just to see if there are other things that come into question. If you don't care about the axon, you don't have to reconstruct it. Likewise, if you don't care about spines, you don't feel that you have to reconstruct them just because you can see them. However, if it's something that you want to include in your analysis, clearly, if you see it, you need to trace it. The other nice thing about Neurolucida is that you can resume tracing at any point. So if initially in your initial analysis and your comparison between structures or cells, you see a difference in the branch structure, which represents which you did initially trace, and that leads you to wonder if there are any differences in the spine density along those branches that you've traced. You can then go and add those spines at a later point. You just have to make sure that you're at the right Z position before you start tracing so that everything stays in registration. Okay. So when you adjust the opacity of the tracing, the color is not as vivid. So I can turn thickness off so that you can see the difference between tracing with and without thickness. Personally, I like to have thickness on so that I can make sure that I'm adjusting the cursor properly as I'm going along. It makes it easy to identify mistakes that I might make as I'm tracing. But if you don't care about thickness, then first of all, you don't need to continually adjust the circular cursor. But if you do, you can keep the tracing settings, the display preferences, set to opaque, and you can see that the color of the resultant branches changes depending on the transparency. So you're seeing as it gets darker, it's just that the branches are shining through and making it look like a darker structure. Okay, so there's been a couple of questions about how to distinguish the Axon versus Dendrites, and I leave that to you. You guys are the experts in your systems, so I don't have any magic tricks for you. It's an anatomical thing, and it'll vary depending on your cell type and what you see and how it's visualized. So I don't have any pat answers for you, unfortunately. Okay, so one quick resumption of, you can see here in the orthogonal view that as I focus up and down, you can see my position in the orthogonal view tracks with what you can see that's going on in the Z meter. And if I were to reconstruct from the cell body, let's pick a cell or a branch that changes a lot in Z. This guy definitely does. So if we trace this one, I'm not going to adjust the thickness just to speed up the tracing. But keeping things in focus, so I'm adjusting manually the focus as I'm moving through the tissue. So I'm focusing with my left hand and tracing in Neurolucida with my right hand. You can see in ortho view how the Z position of this white branch is relative to the yellow and blue branches that I've already traced. And you can see, again, as I finish a branch ending, when I get to the distal tip, once I hit ending, it brings me to a node that I haven't yet traced the branch from and brings me to the right Z position. If it has trouble moving to that position, you should check your calibration. Okay, so if you want to take a break, if you've done all the tracing that you can handle at the microscope, either you need to attend to something else in the lab or someone else needs to use the microscope, I recommend that you finish the branch that you're working on, just so that you don't have to come back to it. It makes alignment a little bit easier. And then you can save the data file. You can save it either as a DAT file or an ASCII file. Generally speaking, it doesn't really matter. I will say that ASCII files tend to manage very heavy data files a little bit easier. So if you've got 20,000 data points or something like that, an ASCII file will load the data a little bit faster. Other than that, there's not much of a difference that I know of. You can take the ASCII file and port it to other programs. The DAT file, not so much. So I default to saving as an ASCII file, but in reality, they're very similar. So we can save the data file. Okay, and let's go back to the presentation so that we can talk about Neurolucida Explorer. So once you've made a reconstruction of the cell that meets your criteria and you're ready to analyze it, you can see that Neurolucida is the program for reconstruction. So you're going to either input your image by an image stack or trace live at the microscope, and you're going to be manually tracing to create a trace model, which you can edit using the new edit toolbar. Inside Neurolucida, you can visualize it with 3D visualization module. And in Neurolucida Explorer, you can analyze the data. Notice that Neurolucida, you can't get very many numbers. So Neurolucida Explorer is where all the analyses are. It comes free with Neurolucida. There's nothing you have to purchase. It is just a better way to break out data analysis from data creation. So one of the typical analyses that a lot of people are interested in is the Scholl analysis. And you can see here that the Scholl analysis is centered on the cell body. And then you will define the radius, the start point of the initial sphere, and then the radii of each successive sphere. And the Scholl analysis will give you the number of intersections for each sphere, each shell, as well as how many nodes were placed within that sphere. The total length is the total length contained within that shell only, and none of the shells that are interior to it. And so from that, this is a very elegant way to take a look at the data to find out the ramifications of your dendrites relative to the soma. Another analysis that is typically used, one quick point about Scholl analysis. If you've got multiple cells in the file, you need to make sure that you select only one before you continue. Branch structure analysis also has just some tremendous analyses available. I can't go into all of them, but if there's something that you want to know about, submit it via the question window or bring it up in the user form and we can address it there. But within the branch structure analysis tab, you have, as you can see, just a number of analyses that are available to you. The segment analysis, as well as neuron summary and each tree and tree totals, I tend to find very helpful. So I'm gonna demonstrate the neuron summary, the tree totals and spines within this aspect of Neurolucida Explorer to give you an idea of what the data looks like. Then another one that I think is really interesting is convex hull analysis. This measures the size of the dendritic field, and the analysis generates a convex polygon by connecting the tips of the distal dendrites and reporting the volume and surface area of that polygon. And so this gives you an idea of how big of a spread is the dendritic field. So before I move to Neurolucida Explorer, let me demonstrate just a couple more things in Neurolucida. In the Edit toolbar, when you select Edit Objects or Select Objects, which you can get to from Edit, Select Objects, the toolbar will appear. This is brand new in version 9. And now you can select multiple objects. And I just want to show a couple of things that are pretty snazzy in version 9. First, you can select individual points. And so now I can select all of the points along a segment as part of a segment. And now I can adjust the thickness globally. So if you have to make thickness adjustments or if you have to move some of the points, whether it's part of a tree here or a different part of a branch, you can just select them all and move them all at the same time. This is pretty helpful, I think. And so if I'm going to move these guys back, I can do so. Okay. The other thing that is very helpful that Neurolucida allows you to do is this concept of sets. And so if you select trees or cells, whatever you select, holding down the Shift key to select different objects, we can do what's called place trees into sets. And this allows us to segregate dendrites, perhaps by ventral and dorsal or some other aspect that makes sense to you. And then we can do analyses on the basis of that to compare one to the other. Okay. So moving to Neurolucida Explorer, I've loaded a data file already. This data file has a number of trees that have been traced, as well as an axon with varicosities, and some spines that have been placed as well. And so I created two sets. I've got some of these sets, some of the dendrites grouped together as ventral dendrites, and then I also grouped some of the dendrites as dorsal dendrites. I did this in Neurolucida. And so now, if I want, I can go to Analysis, Branch Structure Analysis, go to Tree Totals, and select Dendrite Totals. And now I have the dendrite totals for only the dorsal dendrites. And now if I select the ventral dendrites, I can segregate the analysis on the basis of the distinction I made in Neurolucida inside the DAT file. And so here, the ventral dendrites have two trees that are quaternary. There's a total of all of these spines, and you can see where they're distributed. And so you can start to make these comparisons between aspects of your cell. If you have multiple cells in the same file, you can just define your sets so that you can do set analysis that represents the entire cell. So each cell would be its own set. I'm going to pause now for any questions about this transition to Neurolucida Explorer. Okay, there's a couple of questions about the Scholl analysis. Erica asks if the Scholl analysis is a 3D measurement. It is 3D. So if you traced if there is three-dimensional information available, the Scholl analysis is a sphere. If you only have 2D information available, that it would basically be planar. Anya had the same question, actually. Chandler asks, if you alter the thickness of the cursor while tracing, when you visualize the final reconstruction, will your model reflect the dendrite thickness of your tracing, or is that thickness selection just something you might do to help you perform an accurate dendrite tracing? Adjusting the thickness of the cursor while tracing allows you to approximate the thickness of the branch as you see it, and you can export that volumetric information. So you're actually collecting data when you do that. It's not solely for visualization purposes, so you're getting a lot of data out of that. It is more time-consuming than simply just placing the cursor and through Z as you're focusing, but the reconstruction is great. There's a question about whether or not you can identify the dendrites and analyze them without identifying the cell body. If you are only interested in the dendrites, you don't actually have to trace the cell body, although I would recommend that you do so. A single simple contour that represents the cell body is often very helpful. The data analysis that you can get is worth the effort, I think. Okay, so if we're interested in dendrograms, which is another way to visualize the information of the reconstruction in a graphical way, just like the Scholl analysis. If we select the entire, let's select the axons. Well, let's not select the axons. Let's do dendrites. And so we can select dendrogram. You select dendrites and hit display, and now you can see that each tree is now colored by the branch. So this yellow representation here is this yellow dendrite here, and so on. Here's the hot pink one that's right next to it, and you can see that the primary branch is 114 microns long. It has two secondary branches. One is 61.8 microns, and the other one is quite long. And if we look at it, you can see that that's the case. And so this is another way to display the data, and this can be a very straightforward way to get a feel for what's going on in the data as a whole. The current cell body in this particular data file is a single contour. We were not interested in this data file in having a volume of the soma, and so since that's the case, a single contour to represent the midpoint of the soma is all that we decided to do. As I said, Neurolucida can give you as much information as you want as long as you're willing to put the time in to reconstruct it. AutoNeuron is our automatic neuron tracing software, and I'll end up doing a webinar on that as well. When you're doing the Scholl analysis, the center of the concentric circles is based on the very midpoint of the soma that is traced. So if you have multiple contours that represent the soma, then it would be the center of all those contours. With one contour, it is the very middle. So you need a soma for that. we want to take a look at neuron summary. If you select neuron summary, I'm just going to do one analysis at a time. So here's a great way to get a feel for what's going on. In this case, we have only dendrites selected here. So you can see here that the quantity of the axon and cell body is 0 because I only have dendrites selected here. But you can see that there are 7 dendrites, there are 13 nodes, and 52 spines that were placed, and then they're broken up according to the classification of the spines. And this was all done with the spine marker bar in Neurolucida. The total length of all the dendrites is a little bit over 2,000 microns. And if you had traced with thickness, the volume measurement would be helpful. So if we select all of them and do the branch structure analysis, now you can see that the cell body has a quantity, the axon has a quantity, and you can see that the axon itself is greatly ramified. It has 87 nodes, 88 endings, with a total length of 5,200 microns. When you have the data that you're interested in, you can export it to Excel. And so you can select multiple analyses, and multiple pages will be created in Excel automatically for you. Okay, so I think we're just about set here. I appreciate all the patience, and more importantly, I appreciate all the questions. This is great. And so what I would like to suggest is that you provide any feedback to me about what you'd like to hear next, as well as what we can do to make these webinars more informative for you. And more importantly, head over to the user forums and provide some of these questions. I will certainly respond to them, and then we can also get some information from your colleagues and peers who are using Neurolucida. So again, thank you very much for your attention. I look forward to hearing from each and every one of you, and be in touch for the next webinar.